because this is a FIP quiz, here's what I do with these in chemistry and physics, and I'll do the same thing for you guys. I will scan this key, I will post it to Classroom. So if you want to use it to review or look at where you differed from what I had, you can go look at the key all by yourself. So if you have a condition that's carried on any of the first 22 pairs and that you have to get 2 copies of the gene to be affected by, it is autosomal. I did have some people who said autosomal. It's an autosomal recessive. There are also autosomal dominants. So that is a condition which is called an autosomal recessive. With autosomal recessives, because they're not on the sex chromosomes, they affect males and females exactly identically. And that means that when we do a Punnett square for it, we don't need to worry about X's and Y's. The Punnett square just uses uppercase and lowercase letters. Now, traditionally, we use something like big N, little n. You are free to use other letters. I had some people use big A, big T, little t are great. Big C, little c are not so good because the uppercase and lowercase are shaped the same way. They're just different sizes, and if you're not careful, you can get them confused. So it's really strongly recommended that you use a letter where the uppercase and lowercase are a whole different shape so you don't screw that up. Like I don't ever use M's, I don't use W's. R's are fine, you know. But N, when we're talking about just generic autosomal recessive disorders, when we're talking about diseases, we usually for some reason use the big N, little n. Okay, so given a cross between 2 carriers, and you had to know that a carrier had 1 copy of the gene, their offspring would look like this. Whoops, I just about screwed it up there. So you got a point for doing that, one of your imaginary points out of the 13 imaginary points. That's part A, so that's a checkbox. For part B, and a lot of people forgot to do the phenotype on this. You had to do both the genotype and the phenotype. So the genotypes, there are, as with all Mendelian traits, 3 possibilities. Big N, big N, big N, little n, little n, little n. So homozygous, dominant, heterozygous, homozygous, recessive. So those are 1 to 2 to 1. You could probably do that in your sleep by now. Whoops, and there I went and did the wrong thing. So the ratio is 1 to 2 to 1. Okay, most people got that. What a lot of people forgot to do was follow that up with the phenotype ratios. So the phenotypes here. The way we very often break these down for Mendelians is, there are 2 phenotypes. So it's fine to say unaffected and affected. And there are 3 offspring who are unaffected and 1 who's affected. But you kind of have to put a little asterisk by that unaffected because 2 of those unaffected individuals are what? Carriers. 2 are carriers. And so I had a few people, especially in the other class, who wrote unaffected. They did it like this. Unaffected, not carrier. Sorry, my writing is bad here. Carrier and affected. So they basically broke it down into 3 different phenotypes. If we're talking about curly hair or freckles, we don't care if you're a carrier for the gene. It's not going to affect the quality of your life. When we're talking about a disease that can shorten your life or make your life really difficult, we tend to care if you are a carrier or not. So we don't necessarily talk about carriers for, a trait like red hair or cowlick or, widow's peak. But we do talk about carriers with diseases, with genetic disorders, with autosomal recessives. Okay, so that is 1 to 2 to 1. It follows the same ratios as the genotype. And I can shrink that so that you can see it all. There. Okay, so those were the first couple of questions for that. But now what we really want to do, if you're sitting down with a couple and you're a genetic counselor, and this couple wants to know, percentage-wise, what are their chances? For part C, we would say, 25% not affected, not a carrier. 50% of their offspring we would expect would be carriers. 25% of their offspring, on average, we would expect would be affected. Now, again, we said it, a bunch of times. Could they have 8 babies, all of whom are super lucky and who are not carriers and are not affected? Yeah, they could. Could they have 8 babies who are super unlucky and all have the disease? Yes, they could. It can happen. So these are just statistical averages. They do not translate, every conception is a new event. These are just the odds at the beginning. Okay, so that's the front page. That's autosomal recessives and doing a monohybrid cross. Let's talk about sex linked. Okay, so starting off, a disorder that's carried on one of the 23rd pair of chromosomes is called a sex-linked disorder. Not a sex disorder, a sex-linked disorder. There are conditions that are carried on the Y chromosome also. Gentlemen, the Y chromosome is a teeny little chromosome. The X chromosome is about 4 times the size of the Y chromosome. So there aren't a lot of genes on the Y chromosome, but there are some genes on the Y chromosome. So you can have conditions which are carried on the Y. Guess who's affected by those predominantly? Males. All the sex-linked disorders predominantly affect males because males don't have a backup X. They basically have 2 single different chromosomes. And so what that does is it puts them at risk for all of these disorders that are sex-linked. Number 4, disorders carried on the X primarily affect males. Sorry, guys. You kind of get the short end of the stick on this one. Haemophilia is one of them. So haemophilia is a disorder where the body mismanufactures a single protein. It is a 1 gene change. It is carried on the X. You've all gotten a cut at some point in your life. When you get a cut, you just let it go, and it stops bleeding eventually. Little things called platelets rush to the site of the injury. They're sticky, they stick together, they slow down the flow of blood, and you don't bleed to death from a paper cut. For hemophiliacs, there's a protein that's mismanufactured, and there's a whole sequence of chemical events that have to happen to initiate clotting. In hemophiliacs, that sequence doesn't happen because there's a protein that's shaped wrong. And so for a hemophiliac, a simple injury, a cut, falling off their bike as a kid, can be fatal because they can bleed to death. It will not stop bleeding. Internal injuries are very risky for hemophiliacs. There are different degrees of severity of hemophilia because there are a couple different genes that can be involved. We're making it real simple, yes, no. And for a very severe hemophilia, even a bruise can be very dangerous because the bruise is just bleeding under your skin. It's broken capillaries under the skin that are releasing blood. For a severe hemophiliac, anything that causes bleeding anywhere, an ulcer, a bruise, is very, very serious. So anyway, that's hemophilia. You were asked to do a cross between a mother who's a carrier and a father who is not affected. So what you have to know is that this is carried on the X chromosome. So there's mom's normal X and there's mom's affected X. And you also had to know that if dad's not affected, he is X big N Y. There is no corresponding gene on the Y chromosome. This is a gene that only exists on the X chromosome. It's only on that piece of DNA. So you had to have the little N's up above. If you didn't have that, you just missed the whole thing. So we look at the genotypes for their offspring, and we can kind of segregate this. Here are their female offspring. The top ones are their female offspring. If we look at the male offspring, whoops, I did that in the wrong color. There we go. X big NY, X little ny. There's the unlucky guy. So their male offspring are these bottom 2 individuals. So I'm going to shrink that. We have to separate the genotypes because there are different genotypes for males and females. The females are just these 2 genotypes up here, and one is not a carrier, one is a carrier. None of them are affected. So we would say that that is a 1-to-1 genotype ratio. For males, if we look at the males, those are just the 2 offspring down here. We have 1 unaffected, 1 affected. Okay? So again, that is also a 1-to-1 ratio. Okay, so for the females, the phenotypes, there are 2 phenotypes for females. There's not a carrier and carrier. You could put affected in there, but the odds against a female being affected are so slim. For a female to be affected, she would have to have an affected father and a carrier mother. Now, plenty of male hemophiliacs do live, long lives and reproduce, have kids. It's possible that a hemophiliac male would coincidentally meet up with a carrier female. I don't know what the incidence of carrier females is, but it's possible. It could happen. It's unlikely, though. A lot of things have to fall into place. So the phenotype ratio for females is 1-to-1, carrier, not carrier. For males, unaffected, 1-to-1, affected. So we would expect what percentages? Well, in this case, 50% of females on average are going to be carriers for the disorder. And when we look at the percentages, we have to remember that for females, we're only looking at these two. So it's going to be 50-50. You can't have 25 or 75. On average, a quarter of their offspring would be expected to be carriers, but the numbers are different for males and females. For males, on average, 50% are affected, but 0 are carriers. Can a male be a carrier for an X-linked disorder? Nope. If he's got the gene, he's affected. There is no way for a male to carry a silent gene for an X-linked disorder. Okay? Unless he's an XXY, which is an unusual male, and probably not a male who's capable of reproduction. So, okay, tomorrow, here's what's going to happen. So a lot of you do need some clarification on this, I'm seeing. Some of you do not. That's awesome. What we're going to do tomorrow is kind of keep moving. We're going to start to talk about crossing more than one trait at a time. Those are dihybrid crosses. Because we need to talk a little bit about gene linkages, because that does appear on the test. So, questions, comments, concerns? Okay.